Okay, all right. So let me begin. So welcome to everyone. I see lots of people already joined, and hopefully more will join. This is, of course, a webinar first of its kind for many, many reasons. The first, of course, is that we want to share excitement about new science and new medicine during an otherwise rather gloomy time when we're all separated. And that's also the subject for today's webinar, trying to link the new genetic vaccines against SARS-CoV-2, right? It's not COVID-19, that's the disease, but the SARS-CoV-2 virus, with also recent advances in human gene therapy, an active field that's been growing and will only continue to grow. And so what I'll speak about is by no means comprehensive. And in some ways, it just connects some of these ideas that I've been exploring when I am not otherwise doing pediatric oncology and cancer biology during my full-time job. And the format of the webinar is also new. As you can see, I'll present some ideas just to get the discussion, but in many ways, the people on this panel are far more expert in these questions. And so that's another way of saying that for all the good things that will come in the next hour, you should thank them and thank yourselves. And for all the bad things, you could criticize me, and definitely do tell me, send me a note about what worked and what didn't, and also about new ideas that you may find exciting, because we may do this again in the future. Okay, so let me introduce our panelists. Sanjay Box is a 7th year, I think, MD PhD student in the TRIAD program, currently doing his clinical training. He studied with Elaine Fuchs, a metabolic control of stem cell fate, for his PhD thesis. Dalton Bond is a 5th year MD PhD student, currently working on his thesis with Luciano Marafini, where he's studying CRISPR-Cas9 bacterial and viral evolution. Yaniv Kazansky is a 4th year MD PhD student studying cancer proteomics for his PhD thesis in my lab. Chris Mason is a geneticist, I think, first and foremost, but as you probably will see from his remarks, a futurist who studied many aspects of genetics from humans to extremophiles, and is about to publish a fascinating new book on human evolution that I'm sure he'll tell us about. He's an associate professor of physiology and biophysics, computational genomics at Weill Cornell. And Michel Sadelain is an immunologist who's been working on human gene therapy for most of his career, including, of course, the successful development of the first chimeric antigen receptor T cells for cancer therapy, which is now FDA approved. He is a professor of immunology at Weill Cornell and Sloan Kettering. And so today's subject is going to meander from SARS-CoV-2 vaccines, the new genetic vaccines that are now approved for emergency use that many of us have been vaccinated with. It's going to cover a bit about selfish genes and the central dogma of molecular biology and a little bit of history of science. It's going to cover the concepts of gene transfer and transposable elements, because that's really what we're talking about as biologists. And of course, the application of these tools for genome engineering and clinical gene therapy that, again, is about to become reality and will transform medicine for the years to come, without doubt. Now, COVID-19, of course, it's impossible now to think about science and medicine without thinking about this. And this is data from New York State on hospitalizations with COVID-19 as of last week. And you could see, of course, that we are in the middle of the second phase, at least in New York State. And maybe if this data is an early indicator and we are optimistic, maybe we've turned the peak and it's on the decline. And maybe with the ongoing vaccination, we will avoid another peak, which, as you know, from infections like this are multiphasic and come every few months or so. So the This virus, of course, is probably a zoonotic virus with a, as of yet, not fully defined animal reservoir, but maybe bats. And it's like many viruses like this. The most famous, of course, is influenza. And we're still actively dealing with active, you would say, transmission of the H5N1, the so-called bird flu, and the H1N1, so-called swine flu, influenza viruses, which are much more daunting as pathogens because of their ability to recombine. And that's really the basis for continued evasion of vaccines by influenza viruses. And luckily, so far as we could tell, coronaviruses like SARS-CoV-2 have much lower mutational rates and are much, at least so far, appear to be much more genetically stable, which makes the prospects of an effective vaccine more likely, though, of course, this is all new science that needs further definition. And you could tell this slide is way out of date. The case rate is much higher than this number. Now, we have to talk a little bit about the molecular biology of human and eukaryotic cells. And this is still a great paper to read from Francis Crick, where more than many decades ago, he coined the central dogma of molecular biology, where before the physical basis for these processes was defined, he hypothesized the transfer of information from DNA to RNA to protein. Of course, we now know that this is transcription and translation. He also hypothesized the existence of DNA-dependent DNA polymerases, which we now know are responsible for replicating DNA, and RNA-dependent RNA polymerases, which can replicate RNA. What's curious about this, of course, is he also proposed a direct connection between DNA and protein. And to be honest, I don't know the exact basis for this proposal, but of course, we are as of yet to find a molecular basis for translating DNA directly into protein. RNA is an obligate intermediate. And he, of course, proposed that the opposite can never be, that protein cannot be translated into RNA or DNA, and that's really true. We have yet to find systems that do that. He also, of course, proposed that proteins cannot propagate themselves, but we now know that that's not entirely correct. And prions, of course, is a form of protein-based inheritance that can propagate their own activity through conformational propagation, not really biosynthesis. And so, of course, in talking about the currently approved vaccines, we're talking about RNA-based vaccines, and the idea is to use recombinant DNA technology to encode a coronavirus protein, in this case, the protein on the surface of the coronavirus that's in part responsible for the binding to human cells and subsequent fusion and infection, to express this coronavirus protein in a recombinant form, in a synthetic form, in a way that would allow this genetic construct to be delivered into human cells, like muscle cells, and then be translated into spike proteins that would then be presented by antigen-presenting cells and lead to both humoral and cellular immunity. And these are the two vaccines that are currently being deployed, the BioNTech and the Moderna vaccines. They are, of course, different, but the architecture is the same. It involves a synthetic RNA that we'll talk about in a second, that encodes, again, this spike synthetic form of the coronavirus spike protein with a synthetic cap that's required for recruitment of the ribosome and initiation of translation, with several RNA elements to stabilize this and promote the stable formation of this ribonuclear protein particle that would be recruited to the ribosome, leading to, again, high levels of turnover translation to produce high levels of the spike RNA. Now, what's, of course, enabled this are advances in synthesis of synthetic RNAs. And now we could do this both by solid phase synthesis and by molecular biologic means using in vitro transcription using recombinant RNA-dependent RNA polymerases. These genes are, of course, engineered to express altered forms of the spike protein that are stabilized in the prefusion conformation, producing an antigen and an immune response that's biased towards this more functional state, as opposed to other conformations that may not be important for infection. The caps actually nowadays in modern RNA vaccines are synthetic. They're not just simple 7-methylguanosine caps. They're synthetic ones that maximize initiation and minimize cellular decapping, which destabilizes the RNA in cells. And I love this aspect of it. The untranslated regions that are used to stabilize the RNA actually derived from the globin gene, from the globin transcript, and that's, of course, a great historic element because sickle cell disease was the first molecular disease to be defined, and it's only appropriate in a way that the globin UTR is being used here for this purpose. And then importantly, and this has been discussed extensively, the RNAs are not composed of typical 4 bases, but instead incorporate modified pseudouridine bases to prevent activation of innate immune signaling. There are 2 main pathways for this. One involves toll-like receptors that recognize short RNAs, including commonly used bases, but not pseudouridine, and the RIG-I system that recognizes double-stranded RNAs, which are present in many viruses. And so these constructs are designed to avoid both TLR activation and RIG-I activation, which would otherwise impair the production and expression of the antigen. And then finally, these are naked RNAs that are charged and large and are not efficiently taken up by cells. So they are formulated in nanoparticles to allow for their stability and intracellular delivery. And that's an important aspect of what we'll talk about. And then finally, of course, adjuvants to potentiate antigen presentation and immunogenicity. And so to come back again to the central dogma, the other thing that you'll notice here, which kind of impacts how we think about RNA-based vaccines and their safety, is, of course, this very interesting dashed line that Crick proposed, the potential for...
Oh, apologies, Alex. Looks like we have a question from George. Okay, George, you're unmuted.
Hi, sorry, I just wondering, so the modified bases like pseudouridine, would they change the translation? Would they impair translation from the ribosome, or ribosome seems to not care much?
Yeah, they are the tRNAs that are being recruited to these pseudouridine containing codons are essentially the same tRNAs, so it doesn't impact tRNA recognition, and so they are treated. The ribosome sees them as bona fide sense coding bases, but they avoid the ligation to TLR. That's the trick, and there's very interesting history behind the science that maybe we could discuss later. But yeah, that's a good point. All right, so then back to this to Crick's idea of this dashed line that he introduced here, the possibility at the time that there may be an RNA-dependent DNA polymerase that would reverse transcribe the RNA into DNA. And that, of course, we now know to be true. And that's another fascinating aspect of molecular biology era, where at the time there was intense interest, not so much in viral biology, though many of the people studying this, like Howard Temin, were, of course, virologists, but in trying to understand how oncogenic viruses that can transform human cells heritably, how they impact that process genetically. And this is still an amazing experiment that David Baltimore did that concomitantly with Howard Timmons' work led to the discovery of reverse transcriptase, where the experiment was he took a viral preparation. This is an oncogenic mouse virus called Rauscher murine leukemia virus, purified viral particles, and asked, do these viral particles have the ability to make DNA, an RNA-dependent catalytic process to make DNA, where he mixed deoxynucleoside triphosphates, DNTPs, at warm temperature and observed key features of enzymatic catalysis, dependence on, of course, temperature, which signifies an energy-dependent process, linearity with the increasing amounts of added reagents, and of course the requirement for DNTPs, and then concluded in this amazing page from his notebook that there is an enzyme. And that is the basis for the discovery of reverse transcriptase, which we now know, and of course was recognized by a Nobel Prize for Temin in Baltimore shortly after that. And that explained how, again, RNA-containing oncogenic viruses could transform human cells in a heritable way, because the RNA in the virus is reverse transcribed into DNA, which is then integrated into the host cells to produce now genetically transformed, transgenic, if you will, host cells. And that actually coincided with a really flourishing time in cancer biology, because as part of this, the field quickly moved from thinking about cancer as an autocrine problem, where cells are cancerous because they inappropriately express growth factors, to thinking about them as an oncogenic problem, where cells are transformed by oncogenic viruses because they express mutant forms of oncogenes. And now, of course, we recognize that all of human cancer is by and large caused by mutant forms of cellular genes that become oncogenic. But all stemming from this, again, work in virology and studying these RNA viruses. Now, of course, reverse transcriptase and its forms in many different organisms span an amazing swath of biology. We talked about how they are present in viruses. We now, for example, recognize that many endogenous human retroviruses, the so-called HERVs, encode active reverse transcriptases in our genomes. And then related to them is a whole class of mobile genetic elements derived from retrotransposons, which you could think of as kind of vestigial retroviruses that are stripped down to the bare necessities that again encode reverse transcriptase that can reverse transcribe their transcripts to produce DNA elements that can be integrated into the genome, which is the basis for their propagation as selfish genetic elements. And these sequences comprise almost half of the human genome by weight. It's kind of a remarkable aspect to human biology that is still mostly undefined. We now know that telomerase in our cells that maintains the ends of the chromosomes does so through a reverse transcriptase. The telomerase enzyme is a reverse transcriptase that makes copies of telomeric repeats. And of course, many of the prokaryotic immune systems, like the CRISPR-Cas system, certain classes of Cas use a reverse transcriptase to, again, integrate viral sequences to target them. So that's a...
Like Cas12 and 13, right? I think some of the more recent ones, the C2, but yeah, there's a lot more predicted that might be out there too, which is interesting. I think it's the tip of the iceberg probably for those.
That's right, and I think so too. And I think in different organisms, as I'll mention in a second, there are lots of different classes of molecular systems that operate on nucleic acids, and this biology has been expanding ever since its discovery 70 years ago, and we'll surely find more. Now, ideas like this about the diversity of molecular biology in misinformed hands raise all kinds of questions. Like, for example, this criticism that the new RNA-based vaccines will modify us genetically, and this is a serious issue for us to deal with, because vaccines are the only way to control infections like this, and we really need good education and understanding to allow people to process this kind of knowledge properly. And are RNA vaccines safe? Of course, we now know that they are safe because safety studies have been done with them, and the safety margin is very wide, and the risk benefit is clearly in favor of vaccination, but we must do more. Now, this, of course, hesitancy about new vaccines for coronavirus is not new, and anti-vaccination movements are as old as the idea of vaccination itself. This is a poster from the 19th century protesting vaccination against smallpox, which is a lethal disease, where people objected to mandatory vaccination against smallpox. And the same has continued through 20th century and today. So these are real thoughts and real feelings that we as scientists and physicians need to confront in our public life. Now, this is
Alex.
Yes.
Benjamin Franklin opposed vaccination in Boston. He regretted later in his life.
Yes, that's right. I think, again, lots of prominent, highly intelligent people did and do. So I think this requires an engagement, and maybe that's could be a subject for our discussion in a few minutes. I think this is a real issue that cannot just be dismissed as ignorance. Let's put it that way. And again, there's a real basis for this type of skepticism. And here's a very, very famous case with poliovirus, which again was a terrible incapacitating disease that caused paralysis in children. And the development of vaccine against poliovirus was another, again, massive advance in medicine. There were, of course, two vaccines, the formaldehyde inactivated vaccine and the live vaccine, or Sabin's vaccine. And after many years of the use for the live vaccine, it was discovered that the cells in which the vaccine or the poliovirus was grown were actually infected independently by an oncogenic virus called SV40 that is a true cancer-causing virus. And several millions of people were exposed inadvertently to SV40 in preparations of poliovirus in the 1950s. And again, this is an amazing paper from Sweet and Hilleman that describes the first observation of this new virus that led to ultimately revision of the whole production method and suspension of Sabin's vaccine, at least temporarily. But so there are real risks and kind of unknowns that we need to be careful about and skeptical and present carefully. Now, coming back to the new RNA vaccine and SARS-CoV-2 itself, could it really integrate into our genome? And again, this is not without scientific basis. Recently, there was a preprint from Rudy Yenish's and Rick Young's lab that explored this idea that there may be evidence for reverse transcription of the coronavirus RNA and its integration into the human genome. And this is based on their observation of chimeric transcripts in cells infected with coronavirus, including patient cells. The chimeric transcripts contain sequences from the human genome and the coronavirus, consistent with the idea that there's been a integration of the virus into the human genome. And to corroborate this, there were findings that coronavirus RNAs can indeed be reverse transcribed in human cells by the reverse transcriptase from human line elements, which is a major class of retro elements in the human genome that are in all our cells, and that this could be integrated. And so this is under active discussion. And of course, I'm sure Rick and Rudy and their labs are actively working on this exact question. And this is some of their data where, again, they are detecting in RNA sequencing, Illumina-based sequencing of infected cells, the presence of these chimeric reads, where the two Illumina reads, paired reads, map both to the human genome and to the coronavirus sequence. And the issue, of course, is that for Illumina-based paired-end sequencing, this is a common feature where such chimeric transcripts form in the process of library preparation and have actually been documented, ectopically as just a natural byproduct of the way the sequencing is done. So in and of itself, the presence of chimeric reads may indicate simply paired-end Illumina sequencing and nothing more than that. And of course, as we all know experimentally, the fact that integration could be induced in cells with ectopic expression and activation of line elements, that's true, but all it means is that it can happen. It doesn't necessarily mean that it does happen in actual real cells. And that's because there has been actually no evidence so far of, for example, true transposition of coronavirus, which requires, again, if it's mediated by reverse transcriptase or the line element, very specific sequence features like target site duplication or target prime reverse transcription. They're kind of characteristic pathognomonic features of the specific enzymatic process that's responsible. And so in the absence of evidence like that, of course, it's difficult to conclude that the conclusion that was reached is actually true. So we'll see, in the next year, I'm sure there will be follow-up to this work, and we'll find out to what extent this actually does happen in real cells. But these are the types of questions that I think we're all confronted with, both as scientists and public citizens. Now, nonetheless, the study of this coronavirus, has been just absolutely amazingly rapid. Lots of studies on the molecular biology of the disease and the viruses. And this is, for example, work from our colleague Jean-Laurent Casanova from Rockefeller that asked what is the basis for life-threatening COVID-19 disease. In other words, most cases are not life-threatening, but there are patients who die. And so they found that a subset of patients actually have inborn defects in a specific form of interferon immunity that prevents them from mounting a proper productive response. And indeed, this is consistent with the idea that many severe life-threatening infections are actually manifestations of an otherwise immune defect. And that kind of is, again, evolutionarily a very satisfying idea that Jean-Laurent has been championing for many years.
And Alex, just to add, this also these predispose to also susceptibility to influenza and other viral infections.
That's right. Yeah, clear evidence that many other infections, bacterial and viral, that are otherwise severe, actually associated with that's right, with underlying defects of all kinds. And
certainly viral, yes.
Yeah, that's right. Yeah, and then likewise, even in individuals without a recognizable genetic predisposition, there's evidence of an abnormal immune response that is acquired in response to the coronavirus infection that, again, impairs proper clearance of the virus and leads to a kind of a state of immune activation. And indeed, I think some of the severe aspects of the disease, like coagulopathy, that are associated with infection are due to the production of autoantibodies that are not directed against the viral antigens, but are directed against our antigens, leading, for example, to coagulopathy. And that's actually the basis, of course, for the new therapy for severe COVID, is to block and modulate this immune response and prevent some of the serious complications of the infection. And then, of course, that's an amazing, at least for me, an amazing link to, again, transposable elements, because these antibodies, these autoantibodies, are normally produced by our immune cells, our B cells, and the production of these antibodies is a product of the recombination process by the RAG1 recombinase that normally recombines the antigen receptor genes, the B cell receptor gene in B cells and the T cell receptor gene in T cells, and produces a kind of a stochastically diversified universe of, in this case, antibodies that have the ability to bind to many different antigens. And what's amazing about this is, and this is work largely led by David Schatz, who, of course, cloned RAG1 when he was working with David Baltimore, is the discovery that RAG1 itself is derived from a transposable element that, as shown in this recent work in the lancelet, is still mobile. So the RAG1 gene encodes an active transposable element that can not only recombine antigen receptor genes, but can mobilize itself. And now why it's doing it in the lancelet, we still don't know, but that's kind of an amazing link between transposable elements, immunity, coronavirus, and an RNA vaccine. Now, there are many genes like this, and this is entirely, fascinating biology. And even in humans, again, in addition to RAG1, which recombines our genomes in immune cells, there is PGBD5, which my lab actively studies, that functions in neurons and in solid tumors. And there are many others. Don Rio, for example, described the FAP9 gene, which is a P element human transposase that's active as a P element transposes human cells. The function is unknown. And there are many others. And this is kind of what's fascinating about biology, of course, for all of us. Now, a little bit about mobile elements, which again, what we're talking about here. And it turns out that they're actually maybe the most universal aspect of all living things. Here is, of course, Haeckel's tree of life that organizes all living things on this evolutionary tree. And we now know, of course, in prokaryotes, transposons of all kinds, including Casposons that gave rise to the CRISPR-Cas system that we'll talk about, are responsible for their evolution. And actually, that prevents their phylogeny. Carl that was his ultimate conclusion, is that he was unable to derive the phylogeny of prokaryotes because they're simply exchanging genetic material too fast and mutating too fast to deduce a phylogenetic relationship. In yeast, the Ti transposons are responsible for the mutation of lots of spontaneous mutants that led to the cloning of many classic molecular biology elements, like the Tata box, for example, was cloned in this way. And in ciliates, which were, of course, a model organism and remain a model organism for study of development because they are single-celled organisms that develop like multi-celled organisms, the PiggyMac transposase is responsible for rearranging their genome during normal development, which is totally in contrast to how, for example, our cells develop by transcription factors. Ciliates develop by genome remodeling. As Francis Crick was studying molecular biology and trying to kind of deduce the dogma, many others were doing the same. And Barbara McClintock was studying the development of plants and discovered that in plants, a major source of developmental control are transposable elements, in this case, the activator, which has actually started this whole field. And I mentioned the P elements, of course, in insects and in RAG1 and PGBD5 in vertebrates. So there's this rich diversity of transposable elements that can move genes through organisms, sometimes as selfish elements like viruses, but sometimes as domesticated, co-opted parts of our bodies, like RAG1 is an essential part of the adaptive immune response. So that's I think what's fascinating about this. And of course, now to gene therapy, because bees do it, birds do it. And of course, humans wanted to, for many, many decades, to use tools like this for medical applications for human gene therapy. And of course, one of the earliest approaches for this was direct microinjection of genes. And that, of course, led to lots of transgenic animals. And I still am amazed by just the ability to do this technically. And these are real photographs from microinjection of sperm into eggs for male infertility, which is still in clinical practice, that for couples trying to conceive where men are infertile, one effective way to do it is to aspirate a sperm into a glass pipette and then microinject it directly into the nucleus of an egg. Now, that of course works, but doesn't have the throughput needed for certainly more widely used gene therapy or for somatic gene therapy. And of course, it does raise all kinds of very important questions about ethics that, again, we should talk about in a little bit. Now, for somatic gene therapy, this has been, again, of interest for a long, long time. And some of the key work was actually done by Michelle, who decided, along with Richard Mulligan and Dave Williams and Ihor Lemishka and Dave Nathan and other colleagues, to re-engineer retroviruses that we said can integrate genetic material into human cells, to re-engineer them in recombinant form for delivery, engineered delivery of genes into human cells. And so in this particular, for example, paper, Michelle and colleagues engineered a retrovirus for the delivery and expression of the human beta globin gene for correction of globin defects, like, for example, thalassemias that are caused by the inadequate production of normal beta globin. And this was in clinical trials and remains, and Michelle maybe will tell us a little bit more about this ongoing work. And recently, actually, just this past week, there was another landmark study, this one by Erica Ezrick and Dave Williams and colleagues, who used viral gene transfer for somatic gene therapy of blood cells from sickle cell disease, which is again caused by a mutant form of the globin gene that polymerizes and damages red cells. Here, the virus encodes an RNA interference construct that interferes with the expression of the transcription factor BCL11A that I'll tell you about in a second. And at the same time, another study, analogous indication, but now delivery of recombinant Cas9 ribonuclear protein particle to edit the BCL11 locus and inactivate it genetically, as opposed to post-transcriptionally. Now, this is, again, fascinating biology that is really key to many advances in molecular biology and gene control. And the fundamental question here has to do with the expression and regulation of normal globin genes that encode, of course, hemoglobin for oxygen carrying in our blood. And in adults, the hemoglobin is composed by alpha and beta chains. But in the embryo, the beta chain uses an altered form called the gamma globin, which has much higher affinity to oxygen and also happens to have much less propensity to oligomerize. And so there is a physiologic switch that controls the locus that encodes the globin to switch the transcription of the gamma locus to the beta locus as we develop. And it turns out that the BCL11A is a transcription factor that is responsible for most of the switching. And what's really special about this, and this is work by Stu Orkin and Vijay Sankaran and Dan Bauer and colleagues, is that it turns out that a single enhancer that's specifically engaged in the transcription of BCL11A only in red cells could be mutated by a specific guide RNA. And this mutation is sufficient to block most of the red cell specific BCL11A expression, leading to re-expression of the gamma globin, which then dilutes the sickle cell mutant globin and prevents sickle formation. Or, of course, in the case of thalassemias, which have inadequate beta globin production, now compensates for this by expression of this otherwise normal but high affinity oxygen carrying gamma globin. So this is therapeutic somatic genome editing to restore proper globin function. And again, the safety in these early studies seems very compelling. And the efficacy is also very compelling, where patients become transfusion independent after this form of somatic gene therapy.
It makes you wonder why do we ever stop using fetal hemoglobin? What are the downsides to fetal hemoglobin?
No, that's a great question, and what argues against any serious defects is that there are individuals who normally express high levels of fetal globin. Originally it was called persistence of fetal globin expression, and it turns out to be due to, in many cases, deletions of this element. And these individuals fail to switch and continue in their adult life with high levels of fetal globin expression. And so far as people have studied, they really suffer no ill effects.
That is correct. And Alex, if I may add, you gave the essence of the answer, but we have MD, PhD, and PhD students listening, I think. The fetal hemoglobin has higher affinity for oxygen, as the fetus has to steal oxygen provided from the mother's hemoglobin, right? But after life, we have this adult hemoglobin. And indeed, subjects with HPFH, this hereditary persistence of fetal hemoglobin, they don't have, cardiac defects or other diseases. They seem to be fine.
Can they tolerate high altitudes well?
I wonder about that.
Because in high altitudes, the curve shifts to the right normally. So there may be some problems with going on ski trips.
No Machu Picchu.
Do you think they'd be worse on the ski trips, or you think, if they can get oxygen and keep it, maybe it'd be...
Well, but the oxygen doesn't get delivered to their...
Well, it makes me wonder also, if any of you done, is this conserved in mice as well? I'm trying to know how far back this goes phylogenetically, this switch for hemoglobin.
The switch, yes.
Oh, yes.
Because if anyone's ever worked in a mouse lab, actually getting newborn pups, if you put them in pure CO2, whereas adults will be knocked right out quickly and die quickly, newborn pups won't. I think this is why. Yeah.
I don't know, but the switch occurs in mice, although it occurs before birth. Yeah. So there's a little difference there.
Yeah.
That's right, yeah. Well, and then just to finish up, kind of to give you a flavor of the kind of potential here, here's again, just off the press, a paper from Dave Liu and colleagues that now uses a recombinant form of Cas9 that I'll show you in a second to not only introduce mutations, but to substitute bases specifically. And in this particular paper, this is purely a preclinical study, just in mice, no human clinical trial here just yet. But they studied Hutchinson's Guilford progeria, which in most individuals is due to just a single substitution. It's a substitution of this cytosine to thymine in the lamin gene, and that produces a splice donor defect that results in the production of a dominant negative form of lamin A that's responsible for the premature aging in these individuals. And so here they used a recombinant base editor to convert this T back into C in somatic tissues and delivered this construct via a recombinant adenovirus through the blood. And remarkably, most of the phenotypes in these mice were corrected. Now, of course, this is all in mice, but it points now to the potential of using similar constructs for somatic genome engineering. And so the way these constructs work is they, again, use a Cas9 endonuclease to target it to a specific sequence of DNA. And this construct is a fusion either with a deaminase and a UGI enzyme or deaminase itself that leads to a sequence-specific replacement, for example, here of adenosine to inosine that in cells combined with the cellular base excision repair leads to a conversion of this base conversion. And in this particular case, a correction of the lamin G mutant to a normal form. And so this opens all kinds of applications for, again, somatic gene therapies. And so that's—
Alex, and as I think you implied it, and somatic modifications of the genome without inducing a double-strand break.
Yes, thank you. Yeah, thank you for clarifying. That's right. Exactly. That's the idea here. That's the idea here. So these are some of the—
Do you recall if they noticed a lot of off-target effects with the base editing?
Yes, that's an important issue. There are certainly the first generation base editors have a very measurable off-target rate. These new ones have a much reduced rate, and this comes to, I guess, the safety, right? The safety issue. And again, that's one of the questions I wanted to discuss is what diseases could not be safely or ethically treated through somatic gene therapy. And so I've left some of these points for discussion here, and I want to open it to everybody else. But these are the kinds of fascinating advances in science and medicine that I think, at least I am very, very excited about.
Yeah, I think was a great slides, Alex. Thanks for the presentation. I think I agree. It's almost the emerging kind of golden era of thinking about somatic therapies that are at such a high resolution and granularity that, it's kind of something, or at least I feel like I've been fantasizing about it since I was a kid, like, oh, what if we could do this? But now it's actually happening. So it's pretty exciting, I'd say. I'll also put one quick, there was a really great blog post that went through every bit of the background on how the Pfizer vaccine was developed and how why they use pseudouridine and other variations they tried over the past 30 years. So I highly recommend this really great blog post on like sort of the source code of the Pfizer vaccine, because it was really speculated for a while as to what RNA modifications they were thinking of using. But it was great when it was released because it really was a great summary of a few decades of work to figure out why it wouldn't be completely degraded as soon as it was injected. And but it took it's kind of like McClintock. It took decades. At one point, McClintock was famously told to just not publish her work and stop talking to people. So she kept working, but she just spent like 2 decades just not telling anybody about it and waited until she had enough data to really is incontrovertible. Same thing kind of happened with the BioNTech team, as they were told that their ideas were crap, but they kept on it for 20 years.
Chris, thanks for recommending that blog. I just want to add that the story of RNA vaccines did start with COVID-19. It started 20 years ago.
Yeah.
And the first trials, were not very successful, and in part there were strong immune reactions to the delivery of RNA. As you told us, double-stranded RNA in the cytoplasm triggers TLR3 and innate immune responses, etc. And so that led to a succession of changes, the ones you mentioned, the pseudouridine, etc. Then the capping, the polyAs were stabilized, and then good lipids were found to cationic lipids to package these RNAs. And so really this was developed largely for cancer immunotherapy. And part of it was that because it's so rapid, right? You can, and the COVID-19 illustrates that beautifully. From the time that the sequence of the spike was available to having an mRNA ready to transfect, I'm not sure. In one account, I think I heard 25 days and the other one was 6 weeks, but whatever it was, it's incredibly rapid. And so in cancer immunotherapy, people have tried to harness this technology to rapidly introduce, so-called neoantigens, exonic sequence bearing mutations, and string them together and rapidly create a vaccine. And so, and BioNTech, amongst others, and Moderna and others, we're pursuing these cancer immunotherapies. And then comes along COVID-19, and these challenges or kinks that Chris was just alluding to, over the last 20 years, had been really worked out in such a way that this vaccine, boom, boom, is done. And look, it's pretty amazing.
Yeah, and the amazing things like, oh, we see a variant. Okay, fine, we'll just do a boost, and then we know the variants, and it's a really beautiful sort of synthesis between the design and then the sequencing and the continual learning of variants and adaptation. It's like as our immune systems adapt, so do our the collective consciousness of humanity to respond to threats. It's
yeah, apropos of this rapidity idea, there was also a recent paper from the BioNTech group that studied a mouse model of autoimmune disease, where the idea was to immunize mice with the antigen that's responsible for the autoimmune disease to induce tolerance. And so, in this study, it was very compelling. And the proposal is that if in humans with various forms of autoimmune disease, for example, multiple sclerosis, if the antigen could be identified for a particular individual, then that patient could be treated with this patient-specific vaccine to induce tolerance. So again, this is a beautiful idea that of course needs to be tested and developed further. But that's, I think, apropos of Michelle's comment, this rapidity enables one to do this now, right? To develop these medicines that are very, very tailored.
Yeah.
With respect to the concept of inducing antigen-specific tolerance, though, just that alone is goes back decades, right?
Yes.
But how to do it has not been worked out. But indeed, these RNAs that perhaps you can use to express proteins in a way that doesn't stimulate a productive response, but rather induces tolerance, may allow to do something that couldn't be done before.
Yeah. But we have here.
We have a question from Josh. I'm going to unmute him.
Hi, everyone. Great talk. Thank you. So I have a couple questions. The first related to the sickle cell study that you mentioned, where they use genetic editing to try and fix it. And so I was just wondering, in that study, exactly what's the delivery mechanism? I guess in a lot of these, when it comes to gene therapy, for the COVID vaccine, it's these nanoparticles, but just thinking about how to deliver the CRISPR-Cas9 or these various editing tools, how people are thinking about doing it, particularly for things that might be, I guess, more systemic or not easily accessible to the cells of interest.
So actually, so what they were attempting to do, so BCL11A is this repressor of this gamma globin chain, right? So it's silenced after birth. Normally you don't re-express it. And here the idea is to de-repress it to compensate for the absence of the beta chain, or to buffer the nasty effect of the mutated sickle beta chain. So this BCL11A transcription factor also regulates events, if you like, in B cell differentiation. However, it turns out that it has this erythroid-specific enhancer, leading, as Alex pointed out earlier, to the induction of BCL11A in maturing erythroblasts. And so they specifically deleted this enhancer. So they use CRISPR-Cas9 for that. Now, I have to say, I don't know the details of the methodology. I would presume that they use the Cas9 protein and their guide RNA, applying it to autologous CD34 positive cells. But I have to say that in those 2 patients that were recently reported, I didn't look at the exact methods, but I assume it's an RNP and guide RNA co-transfection in
Right.
CD34.
That's right. Electroporation, yeah.
Electroporation.
Ex vivo
Electroporation,
electroporation.
yes.
Yes, exactly. Yeah.
And the post-transcriptional one was a lentiviral, also of CD34 positive cells.
That's right.
Erythroid specific.
Yes, the other study, yes, used an shRNA, right?
That's
Encoded by through a lentiviral vector, right?
I think it raises an interesting point of, in the hematopoietic system, we can purify progenitors very easily for these types of things, but thinking about solid tissues, it becomes a little bit more complicated. But like, I think 2 years ago now, Michel DeLuca's lab published this case report where they were basically able to regenerate entire human epidermis for a genetic skin disorder by like ex vivo transduction. And so, I think even for solid tissues, these types of ex vivo approaches where you can expand cells is picking up, although I think it's still lagging in solid tissues a little bit.
Yes, and generally speaking, these are just, as Chris said, amazing technologies ushering in a golden age, as he said. I wish I were starting now rather than when I actually did. When you actually had to fight people or by the way, face the scorn of people telling you that what you were doing was outrageous and dangerous. But anyway, back to what you were just saying, there's this beauty in the elegance of how these biological tools are co-opted, all these transposable elements and other things, and transformed into medical surgical knives, if you like. But one common challenge in many of these is at the end, delivery. How do you get it in the right cell? And that has been pretty, for the past years, you work out so many challenges, and then you reach that ultimate one. How do I get it into the right cell? And that's why ex vivo strategies were the logical, historical beginning of this, and they continue to pave the way because, again, obviously, the cell is there in your tube, right? But in vivo, that will be the next big challenge. But I'm confident, But that is a big hurdle, in vivo delivery.
Yeah, and getting the tropism of delivery. I mean, clearly our bodies manage tropism just fine. We manage to have exosome mediated communication or hormones or other specificity transcription factors that have certain cells do certain things at certain times. So I think as the more we learn about, work from, let's say, the human cell S, or even just the erythrocyte specific enhancer, just knowing what all cell type specific enhancers exist, which that map's being built out now. I think as we flesh out more of that, and hopefully it'll be easier, but it's kind of like learning to do how our bodies already do it, but with just that same specificity. But I think it'll come.
Yeah, I think one clear task for us is we need to bring new fields together. For example, I think virologists should be brought together with gene therapists, if you will, because one, to answer Josh's question directly, one clear path is to, again, re-engineer viruses for delivery in a safe and effective way. And so this requires, kind of expert knowledge in virology. And the other, I think, field that needs to be brought together is material science. I mean, people have been working on this for a long time, as Michelle alluded to, these advances in sort of lipid formulation and a particle formulation enabled the BioNTech and Moderna vaccines, but so much more could be done in terms of rational engineering of particles and their selective binding to specific cells and internalization and release and delivery. So I think that's clearly an area. So for physically minded people, this is I think a great thing to work on.
Just since we're talking mostly to students, there's a student of mine, but who was a Cornell student, therefore, who worked on CAR T cells in my lab. He then went to MIT as a postdoc to learn material sciences with Daryl Irvine, and then he started his own lab at the Hutch now in Seattle. And what he is doing is aiming to introduce the CAR, these receptors for antigen, directly in T cells in vivo, and trying to bypass this whole requirement to collect T cells from a patient, manipulate them in the lab, and then reinfuse them. There's a long way to go. Having said that, in mouse models, it's showing some signs of efficacy. As we just said in the delivery, so you can deliver to the T cells. Now it's only a few T cells, you don't control what kind of T cells there are, etc. But it's also true that he, and nobody else who's attempting this at this time, can deliver the gene only to T cells. Mm-hmm. It goes to other places too, all right? So there's a lot of good work to be done here.
Yeah, and it's very, I'm being a bit solipsistic by putting a link to my own book in the chat box, but I have a whole chapter mostly on CAR T and CAR NK engineering in that book, because I view it as something that is on the cutting edge now, but if it became standard, the idea of having tweaked your enhanced immune systems would just make a lot of sense for on Earth, but then eventually you could think about, would you need it on faraway planets? And I guess, Alex, you said I'm a futurist. I think I'm just a present day thinker who's just looking ahead. It's just a question of degree, not of type. We're all future thinkers, but I just I feel like I'm just looking a little bit more, and everyone else can do the same thing, I'd say. But I think it's there's some cases where you could say you're ethically bound to give genetic manipulation or enhancements, and one of them could be to enable people who are infertile to have kids, where otherwise you have to tell them they're not allowed to. But if we have the technology to, you could say they would have the ethical right to do some somatic therapies, even to allow themselves to have kids. or for sending people to a faraway planet where they might be completely irradiated and not be able to survive. And if you have the technology to protect them, would you be ethically bound to use it? Probably yes, I would say.
Well, can I ask a question? So again, we're not talking about germ cell gene therapy. We're talking about somatic gene therapy. So are there diseases that could not be safely or ethically treated?
I'm not going to take the ethical aspect right away, but let's first start with
Safety.
another foundation of medical experimentation, if you want to call it that way, and that's the concept of risk benefit. Okay, and so it's actually not surprising that even maybe these RNA vaccines are being tested first in cancer patients. There's a risk benefit aspect to it. And once you establish feasibility, but more importantly, even safety, then I would say a process can sort of trickle down to diseases or where there is perhaps which would not have been acceptable on the basis of unknown risks, which now become tractable.
Again, so there, I guess if there is a way to, I mean, I guess anything that, if it's safe, I mean, it's like the challenge with a lot of therapies is we wait till people are in stage 3 or 4 cancers before they get some of the more experimental therapies because of that risk benefit analysis, that at that point, the risk of dying is too close. And so you'll try something a little bit on the fringe, but, you know, I think as we do more, I mean, even some of the CRISPR therapies, there's always worry about off-target effects, and prime editing might make it better, but it is, those are, things we have to check, and we have very limited knowledge of how good they really are. So ideally we do something like the Framingham Heart Study, but we do it for the next 50 years for a bunch of people who get therapies and see how they do, and then their kids do. But no one's going to fund a 50-year study, really, not yet.
I think another related question that Ushma Neal brought up now, and she's been trying to champion an intervention that clearly is beneficial, which is HPV vaccine for the prevention of oropharyngeal and cervical cancer. How do we engage with the public to spread these ideas, particularly for very, very complex things like this, or even for simpler ones like the HPV vaccine? And I think that's a key issue that we need to confront.
And I think, Ushma, the program she's talking about is specifically for communities of color. And I think that that's sort of a major thing that we need to consider, that, it's not just people who are skeptical anti-vaxxers or very, I think there's real mistrust with medicine for good reasons amongst a lot of people of color. And I think that we need to sort of really grapple as physicians and scientists with how to better communicate and build trust with people that, frankly, our establishment has sort of neglected and exploited over the years.
Yeah, that's right. And I think, sickle cell disease will be a real opportunity for this, because it's now clear that we will have a safe and effective, some form of gene therapy for sickle cell disease, which of course can be curative and definitive. But again, how to communicate this and engage this community, I think, will be a real test of this, and an opportunity, I think, to do something really, really transformative.
If I can sort of piggyback off of Sanjay's points, kind of as all of these therapies gradually come online, and in addition to what Alex talked about, there's things like antisense oligos and nusinersen for SMA, for example. In addition to the scientific and mechanistic questions, a lot of these therapies, of course, come with a very high price tag. I think nusinersen is something like $700,000 a year treatments. And, we already have vaccine hesitancy, but of course, once you market a therapy as being sort of a gene therapy or a gene transfer therapy, you can imagine that there would be even more potential hesitancy to engage with it. So do you think that the main kind of obstacles for these modalities becoming widespread modalities are scientific, and we kind of have to just wait for the clinical trials to happen, or are they financial or systemic or payment based?
I'm happy to take a first stab, but others may want to comment on this. Indeed, all of these emerging, sophisticated, cutting-edge medicines are costly. Part of it is the actual cost of production. You know, if you handle cells, you know, there's a lot of in-process testing. There's a lot of biosafety testing. You need a specialized facility, trained personnel, and all kinds of procedures in place, and that all adds up to the cost. On the other hand, yeah, the price that some of these companies have charged initially is quite baffling. Now, they try to justify it by saying, well, if we're saving these years of treatment where you would have received, you know, X, Y, and Z and gotten all these complications, and perhaps for some of these diseases and a bone marrow transplant, which costs, you know, $300,000-400,000, and this and that, we're saving you all this money with this one shot. And therefore, since over 20 years, you would have cost, you know, $3 million to the system. If we only charge $1 million for this infusion, that's cheap, isn't it? Okay, so this is a certain logic that's out there. Having said that, I think everybody agrees, or not, maybe not everybody, but most agree that these high prices are not, you know, sustainable. And so I think we have to hope that through competition, through improvements of the technology, possibly through regulation and government intervention, that you know, more acceptable prices will come to be. You know, these Moderna and Pfizer vaccines have been incredible because they became available so fast, but they are expensive. And they require all these freezers to store, etc., right? And they are expensive. So the speed did come with a price. And perhaps there will be, after the urgency of the pandemic, I don't know if this will happen, but a second wave of more conventional vaccines with a lower cost that will come, that will essentially take over, right? That we have this high-tech, expensive, rapid response strategy while the more conventional vaccines come around. You know, the conventional vaccines required express proteins and then purify proteins, etc. Here, you plop your sequence in your mRNA, you know, you have your little cassette there, you know how to cap the mRNA, and boom, you move real fast. So anyway, I think it's, we have to realize that these amazing vaccines came to be very quickly based on 20 years of research leading to the state of the art. And it's amazing and wonderful that they are here, but they are very expensive.
Well, there is one question in the panel, which I think is great, is does chemical RNA synthesis have a lower mistake rate than viral RNA polymerase? And could we intentionally increase the error rate of the spike protein during synthesis to match viral tendency? For example, from the known mutation directory from antigenic shift you could get from GIS-aid and broaden antibody recognition. I think conceptually that's a great idea, because you basically be trying to do informatically what our immune system does natively, which is to get a repertoire built in, but before you see it. It's interesting. And you could even have the, you know, look for where the most mutations are occurring during any given 6-month period, and then redo the vaccine to focus on those areas. So yeah, I think you should send an email to Moderna and Pfizer right now. We should say, maybe they're doing it now. I mean, well, because they're doing it in a responsive way. They're looking at what the mutations are and thinking about responding it. But it's the next logical step to say, well, what if we could see where the mutations are starting to bubble up, and then go to those viral selection sites and then adapt in advance and get all around them. So yeah. I like it.
Yeah, there is a category of RNA that you can look up that's called a self-amplifying mRNA, and that allows to amplify in the cell the RNA. So there, I think there's something along the lines of what this question was about that's already brewing.
Right, right. Yeah, and the idea, of course, there is obviously one needs to document safety, that such a pool of antigens would be safe and not lead to autoimmunity and so on. But the issue there is, I think what Michelle is alluding to, is that there needs to be a sufficient level of expression of any one element to produce a sufficiently active response to that antigen, so that one can't just dilute the pool 1,000-fold and compromise the immunization to all of them. One needs to have sufficient levels of expression. But in principle, it's an idea. It would be kind of a polyvalent, right? This would be analogous to a polyvalent vaccine. Yeah.
In cancer immunotherapy, what some groups have done already at the NCI, for example, is produce RNAs that encode 20 putative epitopes based on sequencing of the patient's tumor, right? Identifying some kind of a mutation in an exon, and for each one generating about a 25 mer, and putting 20 in a row, and then a cap in front, a UTR, a poly A behind, and immunizing that way. These multi-epitopes in personalized fashion. It's very exciting. So far, they don't work very well.
But conceptually, they're a great idea. Yeah. Huh.
Good. Anybody else with questions or ideas, either relating to this or more tangential? There is no bad question.
This may be a little bit of an outlandish one, but going back to the issues of trust and safety, especially in the long term, as you're introducing exogenous RNA into human cells, we're gradually finding out that aberrant RNA can have aberrant results in how the cell organizes itself. I think with several repeat expansion disorders, aberrant RNAs can sort of alter the internal structure of a cell. They can result in aberrant base separation. So do you think that there's any kind of risk of, when you introduce these foreign RNAs, are there long-term toxicities that aren't being captured by current clinical trials, which may, years or decades down the line, result in some kind of, I don't want to say neurodegenerative type disease, but something that we can't predict yet?
I don't know if Chris or Alex can comment to this. Of course, this is, it's thought to be a transient effect, right? When one piece of data that I do know of is when cars are engineering with mRNA, they express the CAR for 5-6 days. By day 7, it's on the way out, and on day 8, it's gone. Now, those are dividing cells, note, right? So this is not data in resting cells, but at the least, it's transient. Having said that, in that window of time, can this, influx of RNA coming from the outside do something like you alluded to? I don't know.
Very good. It's possible, and also not just the RNA, but also there's a lot of RNA modifications. Some of the work from Lee et al. In the Cell paper showed that the SARS-CoV-2 genome has RNA modifications. In fact, every RNA virus we've looked at to date has some degree of RNA modification. So I'm really excited that the RNA mods field, which was kind of fringe in the 70s and 80s and then disappeared, now is back with a big applause and efficacy. So I think I wouldn't just think about the sequence, but just like an epigenetics, you think about the sequence and how it's packaged and modified. Same thing with the RNA. It might just be the sequence as well as its modifications that could both lead to some dysregulation, which is
but I think I understand your point, and in the case of this vaccine, of course, the goal is kind of unidimensional. It's just to produce an antigen and have it presented, and the fate of the cell in which it happened could be terminal, that those muscle cells could die off and they'll be replaced. But you're right, in other tissues where the goal is different, for example, the brain, where those cells persist and are part of active circuits, I think the outcome is very different. So you're right, there's it needs to be considered.
Yeah, it's really interesting.
Okay, well, I enjoyed it. I hope everybody else did too. And I want to thank to Yaniv and Sanjay and Dalton and Chris and Michelle for entertaining us. And I want to thank everybody else, almost 100 people who joined. And let's carry on. Hopefully we'll see each other soon in person.
Alex, thank you. This was a great idea.
Yeah, it was fantastic. Thank you so much.
Thank you.